Welcome to our crash course on the nervous system. So the nervous system comprises of over 100 billion nerve cells. Wow, that's a lot. But what exactly is a nervous system? A nervous system is a complex circuit of neurons and supporting cells. Only known organisms to not have one are sponges. Now, not all nervous systems are identical. They're able to be differentiated by neurons, organizations, or circuits. So organisms have these things called nerve nets, which are essentially interconnected neurons, as you can see in the picture of the hydra. More complex organisms have these as well as nerves, which are a bundle of fibers essentially. They also allow for more complex movements to occur. These nerves arose through cephalization, in which the neurons clustered in the brain near the interior end in animals with elongated bilaterally symmetrical bodies. Now that we know what the nervous system consists of, we can actually break it down into two subsystems. The central nervous system, in its simplest form, comprises of a small brain and longitudinal nerve cords. While obviously more complex organisms such as annelids and arthropods need more complex nervous systems, so they have more complex brains and ventral nerve cords. These nerve cords comprise of ganglia, which are segmentally arranged cluster of neurons. There you have it, the central nervous system. Now, the other subsystem is the peripheral nervous system, which connects the central nervous system with the rest of the body using these nerves and ganglia. So now that you guys know the basic structure of the nervous system, let's talk about its roles. The nervous system's main job is information processing, which is done in three phases: sensory input, integration, and motor output, each of which is carried out by a specialized portion of the neuron. Sensory input is handled by sensory neurons, which are otherwise known as afferent neurons. They transmit info from sensors that detect external stimuli such as light, sound, touch, etc., and internal coordinations as well, such as blood pressure, CO2 level, muscle tension, to properly maintain homeostasis. This signal is then sent to the central nervous system where interneurons integrate, meaning they analyze and interpret the signal. They then relay this to the motor or afferent neurons, leaving the central nervous system in the form of a motor output. One thing to be aware of though is that reflexes, like your hand jerking back when you touch an extremely hot item, it doesn't travel through the brain, so it gives a much faster response, providing the survival edge when time is everything. Now we can discuss the specific parts of a neuron. The neuron shown below is a motor neuron. Within the cell is the nucleus and most of its organelles. They have special crystalline masses called dendrites which catch the signal. Before this occurs, it is said to be in a presynaptic state. However, once the signal comes, it sends the signal in the form of a neurotransmitter down the axon hillock, which is the area where the axon joins the cell body. The neurotransmitter travels down this axon until it reaches the synaptic terminals. So now at the terminals, the neurotransmitter is exocytosized at the synaptic gap between the terminals and the next neuron's dendrites in the form of a synapse. And then this process continues down the postsynaptic cell until the signal reaches a motor neuron connected to a muscle cell, which then triggers a response such as a contraction. So glia or supporting cells help maintain neuron structures and health. Astrocytes regulate extracellular concentrations of ions and neurotransmitters, and they even form blood-brain barriers, which are tight junctions which restrict the passage of most substances into the central nervous system, allowing for the extracellular central nervous system environment to be controlled. And these can also act as stem cells. So radioglia form in an embryo in the shape of tracts. So as you can see in the picture, newly formed neurons migrate from a structure that gives rise to the central nervous system called a neural tube, and these can also act as stem cells. And lastly, Schwann cells form the myelin sheath around axons of many vertebrate neurons. Now, these are highly essential, and they're made up of lipid, which is a good insulator, allowing for the signal to travel faster down the axon. And then the oligodendrocytes support and produce this myelin.
A concentration gradient is created when the concentration changes going from inside of a cell to outside. To find the concentration gradient, you divide the outside concentration by the inside concentration. Inside of our cells, the sodium-potassium pump is what maintains the concentration gradients of sodium and potassium ions. The presence of these gradients are what resting potential depends on. But in order to understand resting potential, we must understand membrane potential. Membrane potential is the electrical potential difference across the plasma membrane. It's typically between negative 60 millivolts and negative 80 millivolts in neurons. The negative sign means that the inside is negative relative to the outside. Now on to resting potential. Resting potential is a membrane potential when there are no signals being transmitted. Resting potential is caused by the diffusion of potassium and sodium ions through ungated channels. I'm going to explain this concept using an artificial membrane with two chambers. The membrane has channels that will only let certain ions diffuse through them, in this case, potassium. When only one ion diffuses, the separation of the charge causes voltage to be created. When potassium chloride is in the chambers, for example, since the ungated channels in this case only allow potassium ions through, it will separate the potassium and chloride ions. Now, there is more of a negative charge in the inner chamber because the chlorine ions remained. However, the negative charge will cause the negative ions to attract positive ions, which will oppose the flow of potassium ions out of the cell. This will create an electrical gradient, and when this is balanced with the resting potential, the cell finds equilibrium. The magnitude of the voltage created by the movement of ions at equilibrium is the equilibrium potential. To find equilibrium potential, the Nernst equation is used. This equation can only be used if the net charge is positive 1, like that of a potassium ion. The equilibrium potential says that to find equilibrium potential, you use this equation. This equation applies to membrane permeable to only one type of ion. In a real mammalian membrane, and not the artificial one used for the example, the membrane has channels for both sodium and potassium ions. Neither of them, however, are at equilibrium. This constant net flow of each ion results in a current. The mammalian membrane is more permeable to potassium ions than it is to sodium ions. Because of this, the resting potential of cells is closer to equilibrium potential of potassium ion than it is to the equilibrium potential of sodium ions. Membrane potential changes from resting potential when the permeability of the membrane for a specific ion changes. Just like ungated channels, our bodies also have gated channels. These channels send the signals of the nervous system. The gated channels open or close in response to 3 stimuli. The first type is the ligand-gated channel. These are found in synapses and respond based on specific chemicals, such as neurotransmitters, binding to the channel. The second type of gated channel is a stretch-gated channel. These open or close when they sense stretch or mechanical deformation in the membrane. The last type is a voltage-gated ion channel. These are mainly found in axons and respond based on change in membrane potential.
The membrane potential of a cell that has gated ion channels may change when responding to the stimuli that open or close channels. Some stimuli are able to trigger what is known as a hyperpolarization. Hyperpolarization is an increase in the magnitude of membrane potential. It can be caused by the opening of gated potassium channels. When these channels are opened, the increase causes a potential to reach approximately negative 92 MV at 37 degrees Celsius. Some stimuli are also able to trigger what is known as depolarization, or a reduction in the magnitude of membrane potential. This can be caused by the opening of the gated sodium plus channels. Is basically the change in membrane potential. The larger the stimulus is, the larger the change in permeability. The larger the change in permeability, the larger the change in membrane potential. Production of action potentials. Action potentials occur whenever a depolarization increases the membrane voltage to a particular value called the threshold. If the depolarization shifts the membrane potential sufficiently, then the result is a massive change in membrane voltage known as an action potential, or the all-or-nothing phenomenon. Once an action potential is triggered, it has a magnitude that is independent to the strength of the triggering stimuli. Action potentials of neurons are very brief. They last for about 1 to 2 milliseconds in duration, therefore resulting in neurons being produced at much higher frequencies. Voltage-gated sodium plus and potassium plus channels are involved in the production of action potential. The downtime when a second action potential cannot be initiated is known as a refractory period, and it sets a limit on the maximum frequency at which action potentials can be generated. The refractory period is caused by inactivation of sodium plus channels The axon hillock is usually the site where an action potential is initiated. Sodium plus inflow during the rising phase creates an electrical current that depolarizes the neighboring region of the axon membrane. The depolarization in the neighboring region is large enough to reach the threshold, causing the action potential to be reinitiated. The process is repeated as the action potential travels the length of the axon. There are 5 steps of an action potential. The first is known as a resting state. Then comes depolarization, followed by rising phase, then falling phase, and finally the last phase known as undershoot. The first step in an action potential is known as the resting state. During this step, the activation gates on the sodium plus and potassium plus channels are closed. Therefore, the membrane's resting potential is maintained. The second step in the action potential sequence is known as depolarization. During this step, a stimulus opens the activation gates on some sodium plus channels. The sodium plus influx through these channels depolarizes the membrane. If the depolarization reaches the threshold, then an action potential is triggered. The next step in the action potential sequence is known as the rising phase of action potential. Depolarization opens the activation gates on most sodium plus channels, while the potassium plus channels activation gates remain closed. Sodium plus influx makes the inside of the membrane positive with respect to the outside. The next step in the action potential sequence is known as the falling phase of action potential. During this phase, 2 things happen. The first is the inactivation gates on most Na plus channels close, blocking the influx of Na plus. And the second is that the activation gates on most K plus channels open, allowing for an influx in K plus. The last step is called undershoot. During this step, both gates of Na plus are closed, but the activation gates on some K plus are still open. As the K plus activation gates close, the inactivation gates open for the Na plus channels, and the membrane returns to the resting state. Conduction speed. The larger the axon's diameter, the faster the conduction. The resistance to the flow of electrical current is inversely proportional to the cross-sectional area of the conductor. The adaptation that enables fast conduction in the absence of a large diameter is the myelin sheath. The myelin sheath is a layer of electrical insulation that surrounds vertebrate axons. Myelin sheaths are produced by 2 types of glia. The first is known as an oligodendrocyte, and it is found in the CNS. The second is a Schwann cell, and it's found in the PNS. During development, specialized glia wrap axons in many layers of the membrane. These layers are mostly lipid, which is a poor conductor of electrical currents. The inward current produced during the rising phase of action potential at a node travels to the next node, where it depolarizes the membrane and regenerates the action potential. A salutary conduction is when the action potential appears to jump along an axon from node to node.
In continuing our discussion on how the nervous system in the body works, we will now look at how neurons communicate with other cells at synapses. There are 2 kinds of synapses, electrical, which only pass current, and chemical, which pass neurotransmitters, and most synapses in the body are made of chemical synapses. To get a better understanding of how this cell communication works, we first need to look at how a presynaptic and a postsynaptic neuron interact with each other. On the left, we can see our presynaptic neuron, and on the right, we can see our postsynaptic neuron. Synaptic vesicles, which are neurotransmitters that are packaged into small vesicles, are created in the presynaptic neuron. They are sent down the axon and towards a postsynaptic neuron, and these 2 will meet at a certain point known as the synaptic cleft. Here we'll be able to get a better understanding of what is actually happening in the synaptic cleft. On the left we can see our presynaptic membrane, and on the right we can see our postsynaptic membrane. The space in between them is known as the synaptic cleft. This is where neurotransmitters will cross across the membrane to the postsynaptic membrane. This is made possible by the use of a voltage-gated calcium ion channel, which is open when action potential decreases in the presynaptic membrane. As a result of elevated calcium ion levels, this causes presynaptic vesicles to open, and as a result, neurotransmitters are released. As neurotransmitters are released, they will open up the ion channels, specifically for potassium and sodium. As these neurotransmitters open specific membranes, especially potassium and sodium, this will allow for the polarization of the postsynaptic neuron. This whole process is known as direct synaptic transmission, and the usual result is a postsynaptic potential, which is a change in the potential of the postsynaptic cell. There are 2 kinds of postsynaptic potentials. The first one being an EPSP, exertory postsynaptic potential, is a depolarization and brings the membrane potential towards the threshold. The second one, the IPSP, inhibitory postsynaptic potential, causes a hyperpolarization and makes the membrane potential move further away from the threshold. A single EPSP is usually not enough to trigger an action potential, but multiple EPSPs can.
In a process known as temporal summation, the terminal branch of a presynaptic neuron will have
Two EPSPs come at it at the simultaneous time. This is enough to trigger an action potential. If in the event that two EPSPs move down different terminal branches, then this can still be added up. Summations also apply to IPSPs as well and can cancel out EPSPs. In an indirect synapse transmission, a neurotransmitter will cause a signal transduction pathway to occur. Certain major neurotransmitters include acetylcholine, certain biogenic amines such as norepinephrine, dopamine, serotonin. Amino acids are also included as GABA, glycine, glutamate, and aspartate, and certain neuropeptides such as substance P and metencaphalin. These transmitters function as either inhibitory or excitatory.
gases are also commonly released as they are used as local
Regulators. And on that note, this will conclude our discussion.
The nervous system is made of two parts, the central nervous system, known as the CNS, and the peripheral nervous system, known as the PNS. The central nervous system is made up of two main parts, the brain and the spinal cord. Surrounding the brain is cerebrospinal fluid, which occupies the subarachnoid space. This spinal fluid serves as a shock absorber to ensure the brain does not get damaged. The brain is also made up of two types of connective tissue, the white matter and the gray matter. The white matter is composed of bundles of myelinated axons, while the gray matter is made up of unmyelinated axons, nuclei, and dendrites. The peripheral nervous system transmits information to and from the central nervous system. It also plays an important role in regulating movement and internal environment of the vertebrae. The peripheral nervous system is made up of the cranial nerves, the spinal nerves, and the ganglia located outside of the central nervous system. The peripheral nervous system has two parts to it, the somatic nervous system and the autonomic nervous system. These two parts work together to maintain homeostasis. The somatic nervous system carries signals to and from the skeletal muscle, mainly in response to external stimuli. It is subject to conscious control. These are things like muscle contractions and reflexes. The other part of the peripheral nervous system is the autonomic nervous system. The autonomic nervous system regulates internal environment by controlling smooth and cardiac muscles and the organs in the digestive, cardiovascular, excretory, and endocrine systems. It is made up of three different divisions, the sympathetic, parasympathetic, and the enteric nervous systems. Activation of the sympathetic nervous system correlates with the arousal and energy generation of the fight or flight response. This includes things such as pupil dilation, salivation inhibition, increased heart rate, dilated bronchs, and more. When the sympathetic and parasympathetic nervous system work on the same organ, they typically have antagonistic effects. Activation of the parasympathetic division generally promotes calming and a return to self-maintenance functions. This is known as rest and digest. The parasympathetic nervous system typically does the opposite of the sympathetic nervous system. Lastly, there is the enteric division. The enteric division consists of a complex network of neurons in the digestive tract, the pancreas, and the gallbladder. The enteric network controls the secretion of these organs as well as the activity in the smooth muscles that produce peristalsis. The embryonic development consists of three parts, the hindbrain, the midbrain, and the forebrain. By the 5th week of embryonic development, 5 secondary brain vesicles have arisen from the original 3, the telencephalon, the diencephalon, the mesencephalon, the myelencephalon, and the metencephalon. Most of these changes occur in the forebrain, which gives rise to the cerebrum. The brainstem consists of 3 parts, the medulla oblongata, the pons, and the midbrain. The medulla oblongata contains centers that control several visceral functions. The pons also takes place in these visceral functions. The midbrain contains centers for the receipt and integration of several types of sensory information. Also located in the brainstem is the reticular formation. A part of the reticular formation is the reticular activating system, which regulates sleep and arousal. Located behind it is the cerebellum. The cerebellum develops from part of the mesencephalon. It is important for coordination and error checking during motor, perceptual, and cognitive functions. It also receives sensory information about the position of the joints and lengthening of muscles. The diencephalon develops into 3 adult brain regions: the epithalamus, the thalamus, and the hypothalamus. The hypothalamus regulates homeostasis and basic survival behaviors, such as feeding, fighting, feeling, and mating. The hypothalamus also regulates the circadian rhythm, the sleep or wake cycle. Animals usually have a biological clock. The biological clock requires external cues to remain synchronized with the environmental cycles. The cerebrum develops from the embryonic telencephalon. The cerebrum has 2 hemispheres, the left and the right, which are connected by a thick bundle of axons called the corpus callosum. Each hemisphere consists of the cerebral cortex overlying white matter and basal nuclei. Basal nuclei contain centers for planning and learning movement and sequences. The 4 lobes of the brain include the frontal lobe, the temporal lobe, the occipital lobe, and the parietal lobe. The primary sensory areas receive and process a specific type of sensory information. Most sensory information is directed via the thalamus to the primary sensory areas. The visual goes to the occipital lobe, the auditory goes to the temporal lobe, somatosensory goes to the parietal lobe, and taste goes to separate sections of the parietal lobe as well. Primary sensory areas send information to association areas nearby. These commands are produced by neurons located in the primary motor cortex. The primary motor cortex is behind the frontal lobe next to the primary somatosensory cortex. Functions segregate to different sides during brain development. The left hemisphere includes language, math, logical operations, serial processing of sequences of information, and focus perception, while the right hemisphere is stronger at pattern recognition, facial recognition, spatial relation, nonverbal thinking, emotional processing, simultaneous processing of many kinds of information, and perceived relationships between images.
The central nervous system cannot fully repair itself if it's damaged, so makes connections with other neurons to make up for the damage. An example is people that have suffered through a stroke. Nerve cell development. During nerve cell development, the nerves reach their target cells by axons elongating. They can elongate from a few micrometers to a meter or even further. These axons do not move in a straight line, but the help of molecular signposts help direct them. Signal molecules will bind to receptors of the plasma membrane of the growth cone, which will trigger a signal transduction pathway. A growth cone is a responsive region at the leading edge of the growing axon. Cell adhesive molecules on the axon's growth cone attach to complementary molecules that serve as a track for the axon to follow. Throughout the development, growing axon expresses different genes. This picture shows the growth of axon toward midline. The first picture is the signal molecules bind to the receptor. The second step, cell adhesive molecules bind to complementary and help direct the growth of the axon. In the third step, axon synthesize receptors split, and this prevents axon from growing back across the midline. Neurons are produced and developed all throughout adulthood. Researchers injected BRDU into mice to label the DNA of dividing cells. It was found that cancer patients were receiving the same marker to monitor tumor growth. Studying their brains, it was found that neurons divided in the hippocampus. It was also shown that mice that were exposed to more stimulation have more neurons in their hippocampus. Mature neurons cannot undergo cell division unless brain neurons come from stem cells. It is difficult to conduct research on stem cells because it is hard to find a source of human stem cells. In 2001, researchers had cultured neural progenitor cells from brains of deceased individuals and surgical tissue samples. Progenitor refers to stem cells from either neurons or glia. This cultured cell divided 30 to 70 times. Some diseases and disorders that affect the nervous system are schizophrenia. It affects roughly around 1% of the population. Schizophrenia is a mental disturbance characterized by psychotic episodes in which patients lose ability to distinguish reality. Some symptoms are hallucination, delusion, blunted emotions, lack of initiative, and many more. The cause of schizophrenia is unknown, but it has a genetic component, which multiple genes are involved. Treatments focus on using dopamine as a neurotransmitter. Depression. There are two forms of depression: bipolar disorder and major depression. Bipolar disorder involves mood swings from high to low, and major depression is having low mood most of the time. Both of these forms of depression have a genetic component. Alzheimer's disease. It's a mental deterioration or dementia characterized by confusion, memory loss, and a variety of other symptoms. With medication, it increases the age for humans to live longer since it progressively gets worse. Neurons in the brain die, which causes shrinkage in the brain. Plaque in the brain triggers the death of neurons. This picture shows a person with Alzheimer's disease, and you can see the plaque formed in the brain. Parkinson's disease. It affects about 1 million people. It's a motor disorder by having difficulty in movement. Patients can experience muscle tremors and poor balance. This disease progressively gets worse as you age. Parkinson's disease is the death of neurons in the midbrain nucleus. These neurons usually release dopamine from their synaptic terminals. There's no known cause, but scientists believe it has a genetic component and environmental factors.